look, I forget once you take his virology class, it's there twice. The only this one, once you take it once, so. Until you take one of my classes, but teaching. Might be in the fall. Taste cheese. Drink wine. I'm just kidding, drink wine. Anyways, nobody took a quiz over the weekend. I'm disappointed in everybody. 0 quiz by people. Hopefully it doesn't bite you in the butt, but you never know. Nobody's freaking out.
There wasn't one over the weekend.
Right, there was no quiz over the weekend, which is why nobody did it. It was good. Everybody followed directions. No one took the non-quiz that I did not post. Just to remind you of some of the things we talked about last week, again, a lot of stuff about substitutions and frame shifts. For today, this is sort of some of the relevant information for reminding you about substitutions and frame shifts. But when we dive back into the substitutions, the idea of synonymous versus non-synonymous are important, and how different changes to different nucleotides of the codon impact the result of being silent, missense, or nonsense. All really important. I think there's also one thing from the previous slides that I failed to mention in lecture, or didn't write a review question about, and that's the fact that I said that synonymous changes are more common than non-synonymous changes. Should be the first question of today's lecture review to remind you that transitions are more common than transversions, but also synonymous changes are more common than non-synonymous changes. All together, the idea is that we keep making the same protein without any changes. At least that's our evolutionary drive, is to keep making the same protein without any changes, so it keeps functioning the same from generation to generation. Also, with substitutions, we want to think about the single nucleotide polymorphisms being important. Frame shifts could also be in the single nucleotide polymorphism, especially if they're a single nucleotide addition. But if there are multiple nucleotide additions, then it can't be a single nucleotide polymorphism. And in particular, when we talk about this next type of mutation, is also going to fall into the category of what we call short tandem repeats. This is called a dynamic mutation, and just like substitution and frame shift, this is caused by a problem with the replication mechanism. And in particular, this problem is called replication slippage, where the DNA polymerase will lose contact, or the DNA loses the contact and it comes back together at a new, different location. Like the frame shift mutation, where we need a run of the same nucleotide to make a frame shift more likely to occur, these dynamic mutations only happen in places where there are already repeats present. And the presence of multiple repeats allows for the polymerase to dissociate, and reassociate, creating the replication slippage problem that we see. You don't need to memorize how the mechanism works, but you definitely need to know the name. It is a replication issue that is kind of similar to a frame shift, but in a specialized case. And instead of adding 1 or 2 or removing 1 or 2, we are increasing the number of repeat sequences by more than 1, more than 2, by several. Sometimes that can go up, and sometimes it can go down, but more likely it goes up than going down. So if you want to look at how this might potentially happen, there's this diagram from the textbook. I don't know if you notice, in this top strand here, it's the same 3 nucleotides repeated all over again. And as you're just doing this, every once in a while, sometimes the strands disassociate from each other. So because there are a lot of these repeats, if it comes apart, maybe it doesn't go back together. Who's ever buttoned their shirt up incorrectly where they skipped a spot? That's just sort of easy to do if you're not paying attention, but also kind of, there's a lot of buttons, sometimes you just lose place. That's sort of the idea here, is you kind of lose place. Now, because there's this sort of extra bit hanging off here, that creates where the mutation is going to occur. And so when these two things are sort of separated, not really separated, but when it's need to resolve this thing, you gotta cut here so you can fill in this little gap here. So that's what it creates the increase of this repeat number. Again, we don't care about the mechanism, just that replication is the problem child in this scenario. There is a particular disease that goes with this particular type of mutation, and since it is more a strange thing than simple sign mutations or disc mutations, we all will learn this type of mutation. And it's associated with a disease called Fragile X syndrome. Do they know anything about Fragile X? As you can see, in Fragile X syndrome, the X chromosome is more likely to break. That's why it's called fragile. Who do you think is more affected by Fragile X syndrome, men or women? They're both. Why would it be men is the answer, what do you think?
Only one X chromosome.
Only one X chromosome. But don't we only have one X chromosome in women? One is inactive, the other is inactive, hopefully. But with Fragile X syndrome, typically you don't have the exact same copy. Because you get typically inherit one thing from one parent, one the other, you get a heterozygous situation. And what makes the chromosome fragile is the number of repeats. If you have a small number of repeats, it is considered a normal X chromosome. If you're a really large number of repeats, that's the fragile state. And if you're somewhere in the middle, it's called the pre-mutation state. And to make your life easier, instead of trying to memorize these ranges, there are some numbers that fall in these ranges that are magnitudes of 10 from each other. So if you simply memorize the numbers 20, 200, and 2000, they have a pretty good appreciation for each of the different states. And these are the numbers I'm going to use on a quiz and exam. I tell you there's this number of repeats, you need to be able to tell me, is this a normal X, a pre-mutation X, or a fragile X, or vice versa. If I tell you it's pre-mutation, you should be able to tell me how many copies of this repeat are there. It is a specific repeat called CGG. That will also be something you'll need to remember when we talk about Fragile X syndrome. It's a trinucleotide, CGG. This repeat also occurs in a gene called FMR1. Not so important, but it is somewhat important as we talk about the effects that are also associated with Fragile X besides X chromosome breaking. It turns out the FMR gene, or FMR1 gene, is important for cognitive development. So if this thing is making a protein that's not as functional as it was, you're going to have a impairment of your cognitive ability, which is fun. Right. Not only can your chromosomes break, but you can have a intelligence issue. So that's kind of an interesting thing. If you increase the number of repeats, also has some other effects. Not just do we make the chromosome more likely to break, by adding more repeats, we get more amino acids in our protein, and that can change the functionality of a protein. The fact that it's a trinucleotide repeat means it's not going to be a frame shift, because we're still getting the exact same frame, just we're adding nucleotides or sets of 3 each time, so we're just getting extra amino acids somewhere, which may or may not cause a problem. But it probably does, because we see these types of conditions show up. One thing that'll be important as we go forward, as we get into the idea of regulation and control of gene expression, is the fact that it is a CGG repeat that is increased, particularly in this particular gene. In bacteria, an A is methylated everywhere there is a GATC sequence. And in bacteria, that methylation is mostly for mismatch repair. What's also there is a protective device, because if your DNA is methylated, you can kill unmethylated DNA and keep your genome consistent or constant. For eukaryotic organisms, we see that a different nucleotide is methylated in a different sequence. And it's CG. And it's the C that's methylated now instead of the A. Now this was before used for mismatch repair. This is used for gene expression control. Last Friday we talked about hypermorphic and hypomorphic effects, where mutations are occurring in the promoter of a gene that can control gene expression. It also turns out that how much of the DNA is methylated also is a signal for changing gene expression. And high methylation means low gene expression. Low methylation means high gene expression. So as you become more methylated, gene expression goes down, or at least transcription will decrease. So as we methylate, you get less transcription, less gene expression. Which of these is going to be more common to occur? Methylation of a C in eukaryotic organisms, or methylation of an A in prokaryotic organisms?
Eukaryotic.
Why would you say eukaryotic?
Because it's easier to pair two than four, right?
The probability of getting two nucleotides next to each other is a lot less than getting four nucleotides next to each other. And so as you look at sort of the use of these things, in mismatch repair, you only use a signal every so once in a while to say, is it the parent or is it the daughter? But when it comes to gene expression, the more sites you have methylated, the more knobs you have to turn, the more numbers you have on your dial. You can methylate a little bit and turn it down, methylate more, turn down more, methylate more, keep turning it down. So we get this gradual sort of gradient of replication or expression by methylation states. That's cool. Being able to regulate things. It's not just off or on, it's several degrees of off or on you can do with. Again, we're going to come back to talking about methylation and how it works to control gene expression, but this is an important case because not only are we making a protein that's slightly less functional, we're also decreasing the expression of that protein at the same time. So more repeats, less functional protein, and less expressed protein in general. So everything sort of compounds onto itself. This next thing is not a class of mutation, but a type of effect that has been very instrumental in our ability to do certain types of genetic tests. That's what's called a conditional mutation. They're called conditional because they're not always lethal. There are places or times or instances where the mutation is not going to have an effect. The most common conditional mutation is the temperature-sensitive mutation. This is something to sort of talk about so you can get just an idea of some of the things we can do with genetics. A lot of times if you break something, you have another way to grow a cell. But some things are called essential genes, what we call housekeeping genes. Something that you need all the time, and if you break it, you're dead, basically. So how do you study things that if you break, you die? But one way is through these conditional mutations. So what we've done, or what's been done over time, is isolating a bunch of these things where they grow at a low temperature, but as you raise the temperature, they stop growing. So the mutation is affecting protein structure only at high temperature because it just can't hold stuff together. It's kind of a cool thing, that sometimes temperature does play a crucial role in the activity of certain things. And if we had a genetics lab, obviously we could test some of these things and see some of these things in action. We just have to take my word for it, but these are sort of fun things to do. Or you can take a look at this picture. I forgot if I included this picture. I always forget if I add pictures or not. Who likes the kitty cat? I just learned my parents' cat went black. That's got a black cat in the house. Not my house, their house. Sad. It's like it's kitties. Okay. Black. I know, I feel sorry for me. I feel sorry for my mom. She doesn't know where to laugh or cry. Because the cat keeps bumping into things. That's weird for a cat to bump into things, it's supposed to be graceful. But it's blind, so it can't help it. But hopefully it'll get better. Why is there a picture of a Siamese cat? Where's it in Lane? What do you think? Or is it not? Do you even know what a Siamese cat is? Who's seen Lady and Tramp? We are Siamese. Why is it different colors? Does anybody know this story? The protein that controls fur color is a temperature sensitive protein. So where's the cat the hottest? Is where the protein is not folded correctly and not making color. Where the cat is the coolest is where the protein can fold properly and affect fur color. So where's the cat the coldest? The edges of the feet and the tail and the ears and the nose and the main body is the hottest part of the cat. If you ever had a cat lay on you, the hottest part is the main body part. It's a pretty cool example of temperature sensitive things in actual action. It's pretty neat. Hopefully you have appreciated that replication is a common cause of mutations. But there are lots of other things that can cause mutations. We call these things mutagens. They can either be the cause of a mutation or they can increase the mutation rate. We don't really care exactly what specifically each of these things are doing, just that we are going to increase mutations or make mutations possible. When it comes to studying mutagens, one of the most important names is the scientist behind the Ames test. Who wants to guess who the scientist was behind the Ames test?
Ames.
Most likely Ames is correct. I forget what his first name is. It's either the guy's name or because of where it happened, which was Iowa State. Which I believe is also the case with not a person's name, but actually it's a location, which is kind of not something you need to worry about. The Ames test is an important test that is used or has been used in the past to identify if something is a mutagen. The Ames test is based off of using bacteria, and we expose the bacteria to a chemical and see if mutations occur. And it doesn't really matter how it works, just that it's able to detect mutagens, or if something is a mutagen. One of the really important things that's added to the Ames test is liver extract. I don't know if you know about the liver, it is a detoxifying organ or breaks things down. And so sometimes the chemical itself is not the mutagen, it's what our body does to that chemical that becomes the mutagen. And so by including liver extract in these assays, we see is it the thing we add or is it a byproduct of that thing that's actually the cause of the mutations. It doesn't really matter what is the cause of mutation. It doesn't matter if it's the thing we are exposed to or the thing that thing is made into. Who thinks it really matters? What is the mutagen? A or what A gets turned into? Anybody? What do you think? Guesses, thoughts? Does it matter? Does it not matter?
What was the question?
Question is, does it matter if chemical A is the mutagen or that what chemical A is broken down into by the liver that is the actual mutagen?
You can get it out before it hits the liver.
I guess possibly you could, but the body, the liver is the place where we get rid of those things. Sometimes it's the kidneys also, but at the end of the day, I would say the answer is no, it doesn't matter. If you're exposed to the chemical, it doesn't matter if it's the chemical or a breakdown process of that chemical, you're still exposed to the mutagen regardless of what happens. So at the end of the day, it doesn't really matter what you're exposed to, as long as you're exposed to the actual mutagen. An Ames test is the way that we identify mutagens. And there's a lot of different types of mutagens. You probably know a few. Does anybody know any mutagens besides the ones that are listed on the slide here? One is in every single ad for this product ever, because the government makes them do it. This thing, cigarettes, and what is the thing in cigarettes that can cause mutations? We call them carcinogens. Does anybody know the common carcinogen in cigarettes? It's not nicotine. Nicotine's not actually bad for you. At least carcinogenically speaking. Otherwise they wouldn't sell nicotine patches. It's the other stuff, it's like tar and other things. Asbestos, not that's in chemicals or cigarettes. But does anybody know any other types of things that can cause cancer besides asbestos?
Aspartame?
Check what?
Aspartame?
Does it?
I don't know. There were theories for a while.
I don't think aspartame is a carcinogen. I wish it were, because it sucks. Then you take it out of all things. Aspartame, yeah. Anyway, so these are the ones that we're gonna look at. 2 types of radiation, and 4 types of chemicals, and then water. Everybody drink up. How many cells did I just kill just then? Water, turns out, is mutagenic, but not mutagenic. It's a weird case. So, I don't know why water is actually listed for these things, but every description of this process requires water to be around. So water is playing some role in this process. It's called depurination. Which nucleotides will be affected by depurination? And let's elaborate, because you've used the word in its own definition, and therefore you got the question wrong. For adenine and guanine. You can't say purines, it says purine in it. But adenines and guanines are going to be the ones that are affected. And in depurination reactions, we are doing something different than we're going to see in all of the other types of mutations. Here, the base is lost, but the sugar stays in the DNA. So we only remove part of the nucleotide and not the whole nucleotide. This creates what's called an apurinic site, because it's lacking the purine at the site. The sugar phosphate backbone is still intact. And that creates an issue that has to be dealt with. If it's not dealt with, DNA polymerase, when it comes to an apurinic site, will add A opposite it. Does this create a mutation? What do you think? Let's think about it for a second, because sometimes it's kind of nice to think about how some things work. You told me that purines are A's and G's, right? What is A base pair 2? G. So we got AT base pair, and then what is base pair to G? C. So if depurination happens, we have, take a look at each one sort of separately. If we remove the A, and the T is still there, and we come along and we make DNA, what do? Yeah, we add an A because A base was T. So that's fine. But if we take away this A, because we depurinated, but this thing is still intact, let's just throw some nucleotides next to it for fun. We're going along, we're replicating DNA. What do we add next? There's nothing to base pair 2, which is why DNA polymerase defaults to add A. So we get an A there. The original base pair was AT, but now it's TA. Which is why this is called a transversion, because the A turned into a pyrimidine. Now you don't need to memorize that or understand how this process works, because that's just way too much effort. And there's other things that I really would like you to remember than how does it actually work. One thing that is interesting and important to think about is a lot of these are only a problem after replication goes through an unfixed error. If the thing gets fixed before anything happens, then no harm, no foul. But if we start replicating our DNA and there's a problem in the DNA, that is when something is going to go wrong. So these apurinic sites are not a problem until we start replicating our DNA, and that's when we get a mutation showing up. Before replication, it's just an error. It's a damaged piece of DNA. Not a mutation, just a damaged piece of DNA. That is something to sort of think about, is mutations and damaged DNA don't mean the same thing. Damaged DNA can lead to mutations if not fixed. So keep that in mind. Just like with replication, a mismatch It's not a mutation, it's just a mismatch. It's not a mutation until we fix the thing incorrectly, which we'll get into as we go forward. So depurination only affects A's and G's, because those are the two purines. It is a spontaneous process, but it is a very rare process. It's not gonna happen very often, but it will happen. And again, the base of the nucleotide is lost, but the sugar phosphate is still in there, so the DNA strand is unbroken. The next few things we'll see the base being modified. The first is called deamination. So here we remove an amino group from any nucleotide that has an amino group on it. And typically these are amino groups that are involved in base pairing. And so if you deaminate a nucleotide, you create some base pairing problems. Again, by itself, not an issue. It's just a damaged piece of DNA, a damaged nucleotide. If it gets fixed, no harm, no foul, everything's fine. But if we start replicating and this deaminated nucleotide is still present, mutations are going to occur. Not that you need to memorize this, but this is going to create a transition. So first thing causes transversions, second thing causes transitions, which is the more likely thing to happen, transitions or transversions. Do you remember this from last week? Transitions are the more likely thing to occur. Which we're going to see as we look at a bunch of these, transition is going to show up a lot more often than transversions. So sort of getting a proof to our concept. I don't care to remember which nucleotides are affected. That's a lot of information that we don't really care about if we're not looking at nucleotide structures. But a lot of them are, and that's what we need to worry about. So again, modifying the nucleotide, which will cause base pairing issues. There'll be a summary at the end which will say all of this. If we remove an amino group, that'll affect base pairing rules. We can also alkylate nucleotides, and this will also affect base pairing rules. For deamination, it requires the presence of a certain chemical called nitrous acid, so nitrous acid. So that's able to sort of pull the nitrogen off the nucleotide. Right here, the common mutagens are what we call ethyl methane sulfonate, which the ethyl group is transferred to the nucleotide to cause this alkylation effect, which you can see here happening. Another one is called nitrogen mustard, which should be mustard gas, if they know what mustard gas is and when it was used and what happened after it was used. Anybody World War? Was it World War I? World War II? Or mustard gas was used? It was so bad, it was outlawed in every country. Can't use this more for anymore. Oh well. Nobody knows any history. I'm gonna make all of you take history. It'll be world history or US history, what do you think? I'll do both. But modifying a base can cause problems with base pairing. And again, it's a damaged nucleotide. Until we start replicating DNA, if we don't fix that damaged nucleotide, that is when the mutation is going to be introduced. Something that is similar to the idea of deamination and alkylation is the idea of base analogs. These are compounds that are not going to destroy or damage nucleotide. They are incorrect nucleotides by themselves. But they look enough like a regular nucleotide that DNA polymerase can incorporate them into DNA during the replication process. And just like a deaminated nucleotide is going to affect base pairing, or an alkylated nucleotide is going to affect base pairing, these base analogs are also going to affect base pairing. So when they're there, they're just a damaged nucleotide. As soon as we start replicating our DNA, that's when we're going to see mutations come into play. And some of them are really cool or really annoying, like 5-bromouracil as an example. It's got two different isomeric forms. Depending on which form is incorporated, it'll base towards one nucleotide or the other nucleotide. So this one is really a fun one to deal with if you have to worry about things. But sometimes it'll base with A, sometimes it'll base with G. But you don't need to memorize this as a specific example. Just as an example of a base analog, not this part over here about fiber or uracil being able to have two different forms. The whole point of these things is we have a non-standard nucleotide, which doesn't form the proper base pair. And as soon as we start replicating in that area, we get a mutation because the wrong nucleotide is inserted opposite the damaged or incorrect nucleotide. Basically, the same thing would happen if we did not perform mismatch repair. We would get a mutation inserted, introduced at some point after replication because of that incorrect nucleotide added. So depurination is its own separate category. We don't change the nucleotide, we simply remove the nucleotide, but only certain nucleotides are removed. Base analogs, deamination, and alkylation are all in the same category. They affect the base pairing because they're non-standard or incorrect nucleotides. Here we have a chemical that can interfere with the topoisomerase activity. And what is interesting about acridine orange is we will typically get Frankship mutations after exposure to this particular chemical. And while you don't need to memorize the part in gray, acridine orange is a 3-ring molecule that kind of mimics the purine pyrimidine thing, which is 3 rings as well. So the space or the length of this molecule is kind of like the space and the length of the nucleotide. So as you cut DNA with topoisomerase, if this thing gets in there, it kind of acts as a base pair. So it just expands the whole thing. So it's not a transition or a transversion. It's definitely a frame shift. We do need to sort of understand that this is a different type of mutation. It's not a substitution, which would be what we would get if we did depurination, deamination, alkylation, or adding a base analog. Those would all be types of substitution changes. Here's our first and only example of something that's going to cause a frame shift. Next we have a UV light. Most people are familiar with UV, and you probably know thymine dimers as the thing that UV light causes. Well, you've been lied to your entire life. Because UV light does not cause thymine dimers, it causes what's called pyrimidine dimers. The cytosines can also dimerize together as well. If you don't know how this happens, if you have two Cs that are right next to each other on the same strand, they can become linked together when exposed to UV light instead of base pairing across the strands. If you try to replicate this area, you get mutations happening. And so I guess we could say they're damaged nucleotides. But we're going to have to change sort of 2 nucleotides. So it's not really a substitution. I'm not sure what you really call it. It's neither substitution. It's not frame shift. But generally speaking, when the DNA polymerase encounters these things, it either guesses or it just skips it and goes on, which is something that we can talk about a little bit later. It is a form of DNA damage in which the nucleotide is changed. So it does fall in the same categories, deamination and alkylation. The nucleotides are physically changed, and that impacts their ability to base pair with other nucleotides during replication. Our last mutagen that we're going to talk about is ionizing radiation, which we have 2 forms that we need to think about most: X-rays and gamma rays. We don't need to care about the other things listed here because those are not that common, but X-rays and gamma rays are things that we're exposed to quite a bit. Ionizing radiation can cause several different types of damage. The most important is what we call double-stranded DNA breaks. Topoisomerase 2 makes a double-stranded DNA break, but it fixes it because it joins those things back together right away. Transposase also creates a double-stranded DNA break. By cutting out DNA and moving it to a location, you gotta cut DNA, but typically it's fixed because it goes back together. When radiation strikes DNA, there's nothing holding the DNA. So once we cut it, they move apart from each other. It's like cutting the string. 2 parts just naturally sort of spring away from each other. This can create a lot of problems if not fixed. And as we'll see, there are two ways in which we can fix this thing. But double-stranded DNA breaks are the most serious type of damage that we can have. And generally speaking, when we fix double-stranded DNA breaks, they're not gonna go back exactly the way they were. But sometimes they can be really close, and sometimes they can be really far away. And as it lists here, things that are possible because of double-stranded DNA breaks, the deletion is actually going to be the one that's most common that we're going to see. These are not deletions in the case of frame shifts, large-scale deletions, so lots of DNA being lost at a particular moment in time. Now, part of this we've already talked about. Think back way back when we talked about telomeres. Does anybody remember what the telomere looks like? Anybody dredging up memories of telomeres? Where are they on the chromosome? Remember that part? And at the end, what is their purpose of that? Remember their purpose?
To stop coding information from being coded.
Well, they keep the DNA chromosome from being degraded, so they keep from losing genetic information. Now, the telomere was a loop, so the part sort of folds back in and creates this loop. But if the loop's not there, there's a thing where the DNA breathes. When the DNA breathes, parts of it can't be digested, right? When you cut the DNA with a double-stranded DNA break, it's also going to breathe. So part of it's going to get chewed away. That's how we're going to get deletions. So the quicker these things are fixed, the less DNA we potentially lose. If there's a way we can fix them, then we can almost go back to the way they were, just might be slightly different. And that'll be the next chapter, which we'll talk about, not really a chapter, but the next section. Which unfortunately I forgot to post because I think I'd get this far, but oh well, what can you do? And as you know, radiation is a chemotherapeutic process. You can expose cells to chemicals as well. But for radiation is pretty good at killing things, and what we see typically with chemotherapy is we're trying to use things that are sort of bursts, where we expose the individual to a chemical for a while, then take it away. So any cells that are dividing really quickly, which is the case for cancer cells, they're dividing more quickly, they're more susceptible to the mutagen, whereas normal cells dividing more slowly are less susceptible to the mutagenic event. So we're going to damage some normal cells, but the hope is the tumor cells will be preferentially or more highly affected and killed regular or compared to the normal human cells. And again, here at the end is just a summary of sort of the issues that we deal with. The one thing that's sort of missing is it doesn't say deamination, it just says nitrous acid, because that's the type of chemical that's involved in that process. And all four of those are in the same category because they are going to affect base pairing that's going on there. We have things that affect frame shifts and also something that breaks the DNA. Sorry, I was more on the ball. Close my head. We do need to think about and use as much time as we have available to us. Now, luckily for us, we've already talked about two different types of error fixing things with code repair. One is mismatch repair, which relies on methylation. We also have proofreading activity. We already have a few things that are going to work. What is the similarity between these two things? When do they work? What is the cell doing when these things are going on? These only function during the replication process. And cells are not always copying their DNA because they're not always undergoing cell division. And so we need ways to fix errors that we encounter, not because of replication, but because of other times. So deamination is something that affects the cells, not when they're copying DNA, it affects them anytime. Same thing for alkylation or radiation. They're going to happen all the time. So we need ways to fix things during replication, but also outside of replication. So we have these basic mechanisms that we need to think about. Now, one of them is not a repair mechanism, but is related to the idea of replication, which would be the first thing we talk about. And so because it's not a repair mechanism, nothing gets fixed. But it is kind of an interesting thing to think about in this last few minutes today. Just remind you about the things that can cause mutations, and you may see this same slide in different formats from chapter to chapter. Don't get hung up on how many are in the list, it's which things are in the list. Because sometimes chemicals and radiations are combined together, sometimes these are combined together, sometimes they're all listed out. You need to understand that replication does cause problems at a very low rate. Mutagens will increase our mutations, and those are small changes. Whereas crossing over and transposition, recombination errors can cause large changes. Again, proofreading activity is an immediate activity done by the polymerase. So we know the wrong nucleotide and we know the right nucleotide. For mass-spec repair, we rely on methylation. We know what the new nucleotide was, so we should assume that that's the wrong nucleotide. So mass-spec repair and proofreading activity know what to fix and what not to fix, based off of either methylation or the process is done during the addition of the nucleotides. But if things are happening outside the replication window, how do we know what is right and what is wrong? The easy answer is a lot of things actually physically damage the nucleotide. That is something that can be noticed or recognized as being a mistake. This particular process will only work if repair has not happened before replication starts or when errors or damage occurs during the replication process. Doesn't really matter, but it's a way that we can continue replication where we don't actually fix anything. Without this process, replication would stop immediately whenever a damaged DNA sequence was encountered. That means no new cells get DNA because replication is completely stopped. During this process, we continue the replication process to make a good piece, make a bad piece. At least one of our daughter cells is going to be okay, the other one's, up shit creek, flat out, because it's getting bad DNA. We're not fixing anything, we're just saying, gotta go. Now the cool thing about this is a form of recombination. It's called here strand exchange. So just to give you an idea of how this process works, if there is a damaged piece of DNA like we see here in orange, we're doing a replication. Replication of the undamaged piece goes very well, but during this DNA damage bypass, the DNA polymerase can physically jump forward to skip the damage and start replicating again. That creates a gap. And the gap is filled, like a lot of things we do in medicine, transplantation. So use the good piece to fill in the gap, which is what we do. We take this strand here and put it right there, and it's going to work because it's the same piece of DNA. We fill in this piece and just keep going. So we still have our damaged strand, but at least we've continued our process. Replication still goes. We're still making at least one good double-strand DNA molecule and one damaged DNA molecule. So one cell's good, the other cell's bad, but 50% ain't bad, I guess. Unless this one cell becomes a cancer cell later on, and you're like, oh, that's a problem. That's the issue to think about. At least with DNA damage bypass. It allows replication to continue when it would normally stop. But again, it doesn't fix anything, so it's not a repair mechanism. It's just a way to allow replication to continue making a good new piece of DNA. This is not a new slide, it's an old slide. Same information as before, telling you the types of damage that we will see. And depending on the type of damage that we're encountering, there are different mechanisms that are involved in fixing each of these different types of things. That is one thing that would be really important going forward, is just remembering what repair mechanism goes with what type of damage. And The first thing that we deal with what's called nucleotide repair. And so these things will need the nucleotide repair system. Depurination, deamination, alkylation, or the presence of base analogs are all things we need to remove because they're wrong. This is caused by UV light. UV radiation causes these pyrimidine dimers, and because there's two nucleotides, we can't use the same mechanism, we need a new mechanism to deal with those things. But just so you know, nucleotide excision repair is going to share a lot of similarities with mismatch repair. So it's not a completely new mechanism, it's just applied in a different context. And then the thing we're going to spend the most time on, which is going to be probably the most confusing things for you ever, is the idea of recombination repair. How do we fix double-stranded DNA breaks? There's two ways. Recombination repair is the more complex, but better way to do it. Non-homologous end joining is the quick and dirty way, but it does create a lot more problems than it actually fixes. So we have these three different repair mechanisms to deal with, not including the things that are associated with the replication process. What do you think will be common between all three of these types of repair mechanisms? What type of activity are we going to see needed for all of these things to occur? Not necessarily exonuclease, but definitely nuclease activity. There's going to be cutting of DNA that has to occur. We've already had an exam on MutH, which cuts DNA. And DNA gyrase, which cuts DNA, transposase that cuts DNA. We're going to see a lot more DNA cutting enzymes going forward. Again, the questions that you'll need to think about is, does it cut both strands or only one strand? Does it cut in one location or two locations? These are the questions that we're going to try to answer as we go forward. I'm trying to set the stage so I don't remind you about this later. I don't want to dive into something that's not going to be finished. That's why I'm wasting your time. Fuck it, let's do it. Here we go. You ready for the two-step process in 2 minutes? 1 step per minute. It's a cool process involving two different enzymes. And depending on which type of damage occurred, sometimes only one enzyme is needed, other times 2 enzymes are needed. So further complicate this idea that we need to talk about. And unfortunately, the clock is not rolled over to 1:49 yet. So I will keep talking. Any good jokes?